So now we have some time for questions, time and discussion. The moderator of this part of the discussion will be Dennis Golan. Dennis Golan comes from Scotland, from the Orkney Islands, and he has been involved in the OYSTERECOVER program, representing the industrial partners of this project. So the floor is now yours.
So Dennis Golan is going to moderate the debate.
Okay, good morning, everyone. That was a very interesting talk. I always find it is quite complex dealing with these genomics is the word. It's a little bit, things seem to be quite, he's dealing with like codes and little bits of clues that tell you something, and hopefully you can piece them together. So please excuse me if I'm not quite up to speed with the entire subject, but I'd like to open, obviously, the floor to everyone here to put some questions to Antonio and Belen. So please, if you would come forward with questions. It's maybe, I think the problem here is that everyone's a little bit scared of sounding really stupid. Okay, here's the stupid one. Okay, so you're not going to be any more stupid than me. So I was, how can we get to these list of genes? I know, I'll start it off. So you have a list of genes. How can you kind of spot the one that's going to do how are you going to spot the gene that's identify the one that's going to make the immune, gives the immunity or the tolerance?
Yeah, these differentially expressed genes are supposed to be participating in the response to this experimental infection that we have created, and they are expressed in the infected samples as opposed to the control samples. Many of them are supposed to be involved in the response, while others are probably not involved in the response or in the tolerance to the disease. But the work that we have to do ahead of us and that we are already carrying out, and which has provided 2 candidates already, in the case that these are not confirmed, there is a long list on which we can continue our investigation and research and continue to find more markers in other sequences, because we have many differentially expressed genes which could be key in the response to this problem. As soon as they are found and we can design markers, we can use them for the selective breeding program with the assistance of selective markers. As Antonio said at the beginning of his talk, once you detect that marker, you can analyze it in different individuals, and for the one which has the variant which provides resistance or tolerance to the disease, you can select that element and you can use it as a reproducing individual to produce the following generation of individuals. That is a very rapid method, and therefore you don't have to wait for years for the selection of reproducing individuals which are healthy. And you are certain, absolutely certain, that the offspring that you're going to have, the spat, will have tolerance or resistance to the disease. Therefore, selection of these kind of individuals is very positive to increase the productivity of these kind of oysters. And it was much better, the result, than in the case of other programs where we just studied whether or not the individuals were resistant. Maybe some individuals resisted because of other reasons, but not because they have variants which provide that tolerance to the disease. Therefore, we're now using markers which are directly related to that kind of resistance or tolerance to the disease. And where we have the certainty, we are absolutely certain that they are transferred or conveyed. With those markers that you have, you still have to check and you have to make a selection. Now we have to test them in different families. Test them in different families and see if those families or those lineages are healthy. Is that right? Yes. We will do that in other experiment with lineages after an experimental infection. We will learn about which strains or lineages by analyzing those markers. But before analyzing them, the key which shows us that those are powerful markers is that the variants that we are finding are found in all the tolerant populations or stocks analyzed, not just in one region, but they appear all over Europe, which is indicative of the fact that that can be a very powerful candidate. If that is confirmed, this might provide very positive results, but still we have to confirm the result. Yes, they are potential candidates and have to be tested, and we have not had the time to test all those candidates. We are covering out analyses which were not initially included in the project, but since we believe are important for the sector, we will do those analyses. Let me say something. The lineages, the strains are already there. The strains were produced some 2 years ago. They were taken to the sea, to the natural beds, and out of the 12 strains that we have, we know which ones had a greater percentage of survival and which ones had a high level of mortality. Therefore, what we still have to do is the analysis, but the samples are already in the laboratory and we can analyze those samples. That is to say, we don't have to start a new experiment. The experiment has been done already. What we have to do is to confirm the presence or the absence of those markers in strains, but the strains themselves. Yes, in the case of France, you obtain certain lineages, but afterwards, in the end, you did not have positive results, if you see what I mean. Do you? So how can we have final results for those specific strains? Because France has a very important market for oysters as well. Yes, the point is that after that test, you could have a similar result. And yes, the advantage that we have is that we have a very long list of elements where we can analyze and we can find, therefore, new markers. Yes, what I mean is that testing, doing all those tests is not easy. I remember what happened in France. They had some positive results in France a few years ago, but later on it was not so positive. Yes, in France, the strategy they used is what I said. They choose the reproducing individuals after the survival of those individuals, using that as a fundamental criteria. In our case, it is different what we do. We have to wait until all the analyses are completed, but the experiment has been done already. That was something which was not initially included in the project, but as I said before, since it was very interesting, we will carry out all those analyses as well. And in a matter of short period of time, we will have confirmation of the results. In case this is not clear, gene or genetic expression that Belén has mentioned before is a genetic expression that is exclusive of hemocytes, the cells involved in the defense mechanism of the oysters. Therefore, we are not targeting just any kind of tissue. We are studying the genetic expression in those cells which are specifically involved in the life of the parasite and which might target the parasite very directly. Yes, you showed 3 groups in the samples analyzed in Europe, 3 differentiated groups. But on the Spanish coast, many populations or stocks have been studied. Are there any differences among them? Yes, Pauline will talk about that later on, but the Galician populations are very similar to each other. After the analysis of the populations, we have found out that all the European populations of oysters are, well, they are not confronted a very important danger, and there is not a great level of differentiation of the stocks in Galicia between one another, even though in other areas, in other regions, there are very important divergences and differences, and this has to be borne in mind for the management of those populations. But in Galicia, all the stocks are very similar, very, very similar in Galicia. Yes, my question is if when we talk about having more efficiency in the selection breeding programs, if you could contribute with some information which might illustrate how much in terms of time the previous experiences have been useful, and how much important they are. Because we know that some of the partners involved in our project, Recover project, have been working on this for a long period of time, many years, working in this direction with traditional methods. And I would like to know how much we might improve our systems by using the new tools that you have shown us to have a very quick recovery of the production of oyster in Europe. Well, in the case of shellfish, more generally speaking, I don't know, but my group is involved in a selection program of turbot, and I think that the benefits obtained is very important. We have a marker for the selection of females, which are the individuals which grow faster and therefore can go to the market much faster. And the problem with turbot is that you don't know whether they are male or female until they are adult. And with this marker, we can learn about this in an earlier stage. Producers collect the females, and therefore they are saving a lot of money. And the gain obtained is very important for them. In other cases, we are not using markers, but we are using selective methods for growth rates of individuals, and the benefits obtained are very important. And therefore, I think that genetics is very important for this kind of studies. Molecular genetics and quantitative genetics are very important, because selective programs are, of course, very important, and they have to reduce the level of consanguinity. So if you don't carry out a very careful follow-up of consanguinity, in a matter of just a few years, the response to selection might be very negative. And therefore, those 2 points are really essential. In the case of shellfish, if we talk about classical, so to say, or traditional programs, because marker-assisted programs, in the area of marker-assisted programs, there is not a generalized experience in this area, not here in Spain, but just all over the world. We are working with this in Australia, for example. Potential markers have been identified in the United States of America. In the case of oysters, of American oysters, there is not a lot of information provided, but with the traditional programs that done with virginic oyster to attack MSX, the programs were initiated in the 1970s, and each generation for the selection had something like 5 years of experience, and each new generation included an increase of survival. And after 5 generations, we had mortalities caused by Acrosporidium melsoni of over 90%, from 90%, such as I said before, to mortalities when they reached the commercial size. We came to mortalities of 20%, from 90% to 20%. So that is a very important difference. It is really different if 90% of your oyster stocks dies, as opposed to having just 20% of all oysters dead, which means that commercially speaking there is a very important difference. But in order to be able to reach that 5th generation, we had to wait for something like 25 years. It is true that we were obtaining better results for each generation, but still you needed 25 years. That is a very important precedent. Another very important precedent, Sarah Cullity may tell us about that, is the precedent of the Atlantic Shellfish Selection Program. Me pregunto, Sarah, si quieres...
I wonder, Sarah, if you would like to explain a little bit about the program of Atlantic Shellfish.
The industrial partners that we've been working with very closely for quite a lot of time now in Ireland are Atlantic Shellfish Limited, and the owners there are David and Tristan Hugh Jones. They came to Ireland in the 1970s and set up a very efficient system in Cork Harbour on the south coast of Ireland. There had been a native fishery there going back to the 1800s, which had been severely depleted and was really pretty much empty of oysters. And over a period of years in the 1970s, they started to restock the natural oyster beds there. And they did this through a mechanism of developing a series of spatting ponds on a small island in the next to the North Channel of Cork Harbour. So this system was quite successful, and the oyster beds very quickly redeveloped and production levels started to increase. And then early in the 1980s, they noticed that some mortalities were occurring in the oyster beds. And I think these may have been occurring for a period of a couple of years before bonamia was diagnosed on the oyster beds. And we don't know exactly how this introduction occurred, but it was the first record of the parasite in Ireland. So the system that David had developed in Cork Harbour at the time involved a series of designated oyster beds in the area. So different aged oysters were on-grown on different parts of the oyster beds. So they were able to designate and identify the age of the oysters and the year the oyster larvae had been produced and were being on-grown. So when the bonamia was introduced, and after a couple of years, mortalities were extremely high on the oyster beds. In the market-sized oysters, they were experiencing up to 90% mortalities. So very quickly, oyster losses were extremely serious in the area. So the strategy David decided to take immediately really was that he would use the survivors of the initial mortalities as the brood stock. So because of the large number of spatting ponds they had, and this method where they were able to identify very specifically the age groups of the oysters that they had, they used the older, larger oysters that had been exposed to the parasite for the longest period of time as a brood stock. They were removed from the oyster beds and put in spatting ponds, and larvae were produced. These were redeployed onto designated oyster beds, and again, over a period of years, these survivors, when they got to market size, were again used as brood stock. And so that's the strategy that has really been taking place since the late 1980s in Cork Harbour, and has really demonstrated a very clear reduction in the impact of bonamia on the oyster beds there.
Thank you. Any other questions on that? One at the back. Just at the back.
Belen, congratulations for your talk. It was very clear, but I'd like to know more about which other aspects. You said there are other genes that might be selected to improve the selection of oysters, and I'd like to know more about those specific aspects. Well, I didn't say genes. I said that some of the resources that we have generated can be used for other breeding programs focused on other kind of characters, but the tool for the genetic variability can be used for that. The genealogical traceability for selection breeding programs can be used, targeted at any other character, and the genes that we have obtained are of immune response to the disease, to bonamia, which means that this could be applied to other diseases, of course, but I think it is difficult to use this list for growth purposes. This experiment was not aimed at studying or the selection of bigger or smaller individuals. That is not the point. We use databases with immune genes. So that is why I mentioned some of the tools that might be useful for that, but in other cases you cannot obtain a great deal of information of that list.
Hello. I just wanted to ask, also you found a SNP which had high occurrence, well, complete occurrence in all the tolerant strains. How variable were your microsatellites at other loci in them, and would that indicate high inbreeding or perhaps selection occurring naturally?
Maybe Manel can answer the question, because the selective depression analysis was performed by him. Manel.
There is not a direct association with the two variants for a SNP, for example. There are a difference of the allele frequencies. Okay, but inside of the locus there are no deviation from Hardy-Weber equilibrium, for example. The test that we are testing we analyze our loci in order to know if the differentiation that you are showing it's higher than expected by neutral loci. Okay, and if you are obtaining values higher than expected by neutrality, it's okay. You can think that this locus is under selection, divergent selection. But as explained Belén, now you have to test on the families, because maybe it's only a effect of the it's only a demographic effect, for example, the distribution of this marker, the frequencies of the marker due to the geographically differences. We are thinking that it's a good marker, but now we have to test and confirm in our families that Antonio it's analyzing and it's performing. I don't know if I'm answering your question. Yeah, it's a really good candidate, really good candidate. And as Belén said, you can find both alleles in all the populations. Now we have to try to test if there are a real association with this tolerance to bonamia.
Any other questions? Maybe I could just ask something. Why are we likely to produce one super resistant stock, or is it more likely for localized solutions for families in different locations that are adapted?
Yes, the problem that we dealt with during these days' debates is that the production of a general, unique stock for the whole region or area analyzed might not be very useful, because oysters are highly adapted to the specific conditions of each natural environment, and therefore they have problems when they are moved from one area to another. Therefore, that means that we have to produce local stocks, specific stocks. And so what is the problem that we have if we try to do this? Well, initially there is no problem, of course, as long as we are constantly analyze the consanguinity in each generation, not to lose genetic variability and losses in selection. Therefore, oyster producers right now, if these markers are actually confirmed, the steps that they should take would be to start or launch that selection plan with one specific stock for a general stock, bearing in mind the three fundamental areas involved, the region, of course, and the area where they are going to be situated. And within the project, we have produced the tools that are needed for the selection program. We have the genealogic traceability tool. We have a list of genes where we can find things in case our data are not confirmed. But if they are confirmed, we should analyze individually potential reproducing individuals to find the genotype for these markers, because we need that the breeding population is varied. We cannot choose randomly reproducers within a specific area. We need to carry out a genetic analysis, and we hope that that stock will be made up of very varied individuals that are genetically different from each other. Once you have identified all those breeding individuals, once you know the genotype of those individuals, you can decide with the help of quantitative methods which crosses you can actually carry out. Another very important thing is that the management of that reproduction activity with genetics is so much easier because the individuals are marked. Very often you don't know how to manage an individual oyster, but you can introduce several oysters, several specimens, because you want to analyze the offspring, the spat of that. And therefore you don't have to put in one base, one female and a male individual, but you can put several individuals in the same vase and wait for the reproduction, and afterwards you can analyze the offspring. After the spat is produced, if the markers are confirmed, we have to learn if the reproducing individuals have the variant that leads to that tolerance or resistance to bonamia. So, for those who have that allele, they will be the one producing the following generation. Therefore, the offspring will have tolerance and resistance to bonamia. So those are the steps that should be followed in the future. But such as we said before, it is absolutely essential to carry out a genetic analysis molecular genetic analysis and quantitative method to carry out a follow-up of this plan and this program. For a sector which is in the hands of many families, very often it might seem to be complicated, but the analysis is very simple, and they will be supported with the participation of specialists. And if oyster producers follow the patterns that we try to convey to them, the benefits will be very important, but always as long as they follow the steps in the system that we are recommending to them.
Jose?
Hi, Belan.
Hi.
I'm just wondering, did you see much variation in the gene expression between your stocks over your 3 different sample time periods, or even within each stock?
No, the list of differentially expressed genes is between lines, times, and the interaction of them, all seen together. So what we have presented are the results, which are statistically significant, are for 2 of the cases, because in one of the cases the oysters were too small, and therefore the amount of RNA that we extracted was very small, and therefore analysis done in that lineage cannot be compared to the one we did with a naive and tolerant lineage. But the list includes everything, includes the whole comparison between all the elements. My name is Jose Fuentes. I am one of the members of this project, and together with Antonio, I was in charge of launching here in Galicia this line of selective breeding programs. I think that all of us present here or participating in the project believe that the only way we can solve the problem of bonamia today is through selective breeding programs. But we believe that we have to target the issue appropriately. We have seen that false expectations have been raised over 40 years of time in the case of oysters. When this information comes to the press, journals, the press do not understand what we say. They convey a message twisted for the population. And the first thing that we have to say is that as experts, we believe this is the only way, selective breeding programs. We do not anticipate any other kind of programs. Second, selective breeding programs take time. We need a lot of time. They cannot be solved overnight, just like that. We need to initiate, therefore, these programs as soon as possible. Selective breeding programs demand 2 things, basically. First, consistency, and second, money. And also, it is very important to have molecular markers, and one of the main components of the OYSTERECOVER program is to have those genetic markers. That is a qualitative change in our direction, because by using these markers, as you know, in the area of genetics, there is a very interesting concept, what we call the generation gain, or the gain for each generation depends a great deal on another concept. Selection intensity. When we can mark those individuals that are more resistant, or choose those that are more resistant as breeding individuals, we are gaining in selection intensity and response to this problem of selection. And therefore, what is important is that this project is opening up the possibility of a new system for selective breeding programs assisted by molecular markers. If all the sectors involved in this issue, including the industrial and the producing sector, researchers and the public governments, local government, national and European authorities are ready to fund these programs, then we will succeed. Otherwise, we will fail. Therefore, we've opened up a new window of opportunities, but we don't have the solution to the problem yet. That is my standpoint. There is another very important issue that Antonio mentioned at the beginning of today's session, and it is that it is essential to improve protocols for the culture of oysters in hatchery, because for the breeding of oysters in hatcheries, because they are very different in different places, in different laboratories, and therefore we don't really know about the breeding systems for oysters in captivity. And that's it. Thank you.
Thank you for your contribution, Jose. Okay, we've got to coffee now for 20 minutes. We can discuss this. Sorry?
Dennis, there's a question.
Sí, hay otra pregunta que viene de la red. Lee Shaston from Belfast. She works at the IVIS project and she's following this seminar online, and she wants to confirm whether or not she has understood correctly when you said that the extraction of DNA samples does not kill the oyster. And actually, how do you do that? Yes, it is absolutely right. It is true. We can do a biopsy. We can take a very small fragment, a very small sample of the gill of the oyster, and this does not necessarily kill the oyster. The oyster actually recovers in a short period of time. This technique has been implemented experimentally already for a long period of time, and basically we use two different routes. One is the sample, taking a sample of a slice of gill, and another system might be to obtain a part of a muzzle. If we are very careful, or careful enough, at least in our laboratory and also in Latin Blood, in France, they are very careful as well with this. We have a long experience in biopsies without killing the individuals where the biopsy is practiced, not just in the case of oysters, but in the case of clams and other shellfish. We puncture the shell, we obtain a small sample of the muzzle, and with time the individual will become healthy, will even cover the shell, the hole in the shell, with a new piece of shell, and therefore the individual will survive, and therefore will have a normal life in the future. Yes, it is absolutely possible.
Thank you. The web broadcast is a wonderful thing. Chair people must beware. So can we break now for coffee for about 15 minutes? We could talk again over this. Thank you.